Thank you for being here today, and thank you for listening to my presentation, my dissertation on the ecology and epizootiology of Montipora white syndrome, a disease of the Hawaiian coral Montipora capitata. So coral reefs are amazing ecosystems. They have high productivity, high biodiversity. They protect our coastal lands from storm damage. They provide food for millions of people, and they drive the tourism and fisheries industry. So they're very worthwhile and very valuable. Unfortunately, coral reefs have been declining over the last several decades, with disease being a major factor in that decline. In 1985, there was only a couple of coral diseases described and very few reports around the world, indicated by the red dots here. But by 2004, not only was there an increase in the number of coral diseases that have been described, but a vast increase in the reports of coral disease in all regions of the tropics. But clearly disease is an emerging threat and needs to be investigated further if we want to continue to preserve the remaining coral reef ecosystems. So the coral disease that I study is tissue loss diseases. And this photo is a good representation of what tissue loss looks like. On the right side of this fragment, you can see the healthy, intact tissue with little polyps here. On the left side of this photo, all that's left is this calcium carbonate skeleton underneath. The tissue has been eaten away, and the skeleton will be colonized by other microorganisms and filamentous algae. So tissue loss diseases are particularly harmful to coral reef ecosystems because this exposed skeleton will eventually be eroded away. And it's the 3D structure provided by the coral animal skeleton that provides the homes and protection for many organisms on the reef. So without the 3D structure, you really alter the whole ecosystem. The coral disease that I study is called Montipora white syndrome. So Montipora refers to the type of coral that's being affected. In this case, Montipora capitata, or the Hawaiian red rice coral. And white syndrome refers to the type of coral disease. In this case, a progressive tissue loss disease. This disease is particularly abundant in Kaneohe Bay, although it's been reported throughout the Hawaiian archipelago. Because of its high abundance in Kaneohe Bay, this is the focus of my studies. So Montipora white syndrome, or as I'm going to abbreviate it on the slides, MWS, can come in two general forms. You can have acute Montipora white syndrome, which is a very fast-moving lesion. It can affect a large proportion of a coral colony and kill a whole coral colony within days to weeks. There's also chronic Montipora white syndrome, which is a slow-moving lesion. Often the lesion itself is smaller, less than 5 centimeters. And this lesion takes months to years to cause full mortality, if it causes full mortality. In order to understand disease processes better, it's important to take a holistic or an epizootiological approach. So there's your fancy word to take home for the day, epizootiology. But this word really is just synonymous with epidemiology in the context of studying human diseases. So an epizootiological approach disease occurs from a complex interaction between three major variables. First, you need a susceptible host with which disease will manifest. You need a pathogen or pathogens to cause the disease, and you need an environment that will either bring these two together or alter the interactions between these two, allowing infection to happen easier. And it's only when these three variables come together under very specific conditions will disease emerge in an animal population. So we already know quite a bit about Montipora white syndrome from all three of these aspects. For example, the susceptible host comes in two color morphs. There is a red color morph shown here on the right side of the graphic, and an orange color morph shown on the left side of this graphic. So these two color morphs are the same species. They exist on the same reefs, as you can see side by side in this photo. But the orange morph has a higher prevalence of Montefiore White syndrome, despite the fact that the red color morph is just more prevalent, or sorry, more abundant on the reef. All right, so this indicates that there's differential susceptibility to Montefiore White syndrome among these two color morphs. So this dichotomy is pretty cool because that means we can now use these two color morphs to study what aspect of the host might give rise to disease resistance in a population. Our lab has discovered 3 bacteria that can cause tissue loss in the lab setting. So one is Vibrio coriolyticus, strain OCN008, Vibrio owensii, OCN002, and a species of Pseudo-Ultramonas that we designate OCN003. So we'll come back to these guys. You don't have to memorize them quite yet. And then from kind of an environment side, Kaneohe Bay has experienced several outbreaks of acute Montefiore White syndrome in the last decade, all of which have happened during the winter rainy months. And 2 of those outbreaks had the highest disease abundance in the southern region of Kaneohe Bay, which has the most urban development, is enclosed, and has other anthropogenic kind of impacts to it. But even though we know all this about Montefiore White syndrome already, a lot of questions still remain. So some of the questions I was interested in asking was, what drives the differential susceptibility to MWS that we see between these two color morphs? How are the corals getting exposed to pathogens in the first place? And what components of the winter conditions affect Montefiore White syndrome infection and maybe led to the outbreaks that we saw? So to answer those questions, I set up 3 objectives. And the first one was to compare coral host defense in terms of their associated bacterial communities. And this is because in humans and in many other animals, bacterial communities are an essential part of health and protecting you against disease. So, and this has been shown to apply to corals as well. I screened several sources of water and animals in various places in Kaneohe Bay to try to search out the pathogens of Montefiore White syndrome. And then I wanted to do some manipulative experiments to start to better understand some aspects of the environment that might be influencing Montefiore White syndrome. So in the case of heavy rain events during the winter season, there's a lot of stressors that can come into the bay: cold temperature, sedimentation, fresh water, lowered salinity. So I wanted to test just to start off with one of these stressors. So first we'll start with my first objective, to compare the bacterial communities associated with both of these color morphs while they're healthy, and then assess how those bacterial communities change once a coral develops Montefiore White syndrome. So hopefully with that kind of comparison, we can get some insight into what's driving the differential susceptibility and maybe kind of better understand what's going on at the microbial level during the disease itself. So I was really interested in studying bacteria. Like I said, they're a really important component of your health because they have 3 major functions in most animals, including corals. So one, the bacteria associated with your tissues can provide a physical barrier to pathogens, like blocking entry and access to tissues. The bacteria on a coral can also make antimicrobials that will limit the growth of their neighbors and potentially limit the growth of pathogens. And they can occupy metabolic niches. So what that means is they can use up valuable resources and nutrients so that pathogenic bacteria can't use those resources to grow and proliferate and then infect. So in order to look at the bacterial communities on Montefiore capitata, I utilized high throughput sequencing of the 16S, or the V3 region of the 16S RNA gene. So I'll walk you through this. So first I went out in the field and I collected some fragments off of colonies that looked completely visually healthy. So I collected fragments from 5 red color morphs and fragments from 5 orange color morphs. I also collected lesions off of colonies that were displaying the slow-moving chronic Montefiore White syndrome. Again, 5 from the red, 5 from the orange morph. And then the same thing, collecting lesions from the fast-moving acute Montefiore White syndrome. 5 from red, 5 from orange. And then I got all these fragments back into the lab. I extracted the DNA from these fragments and amplified the 16S gene. So the 16S rRNA gene is present in all bacteria, and it's useful for identification. I sent those 16S gene copies to sequencing and got over 500,000 sequences back. I conducted bioinformatic and statistical analysis in a program called SHINE. If you want more details on this, you can bug me later. And then I identified the bacteria in my samples, and then use the data so we can visualize what the community structure looks like and what's happening in our samples. So before we dig down into my data, I kind of want to go through an example of how a common way of visualizing bacterial community structure. And we do this via principal components analysis plots. So in this simple example, say we have 3 samples. These might be 3 different coral samples. And the bacteria on those samples are represented by these letters. So sample 1 has a bacterial community that's completely different than sample 3 in this example. And then sample 2 has bacteria that are shared with both 1 and 3. So if you were to put this on a plot that kind of takes into account the whole community, if we plot sample 1 down here on the corner, then sample 3 would have to be very far away. They don't share anything. And then sample 2 would be somewhere in the middle because it shares bacteria with both this one and this one. So as we look at these principal component plots, the thing to take away from this is that each dot on the graph represents a sample. The bacterial community is within that sample. Oh, sorry. And the closer the dots are together, the more similar the bacterial communities in that sample is. All right, so now we can jump into my data. So let's first look into just the healthy red and orange color morphs. I found that these 2 color morphs had very different bacterial communities. So if you use a weighted metric here on the left, which takes into account what bacteria are present and how abundant they are, you can see that there is a divide between my red color morph samples and the orange color morph samples. If you look at a metric that is unweighted, so it's only looking at the bacteria that are present or not, there's an even bigger divide between the red color morphs and the orange color morphs. In fact, the red color morphs on this graph are very close together. So they have a bacterial community that's very similar and is probably very tightly conserved. In contrast, the orange color morph has much more variability in the types of bacteria that associate with it. So if we look at how these communities are different, I plotted the most abundant bacterial genera, and they're plotted by relative abundance. And we can see for the red color morph, the dominant genera are pretty well evenly distributed. In contrast, the orange morph is heavily dominated by Vibrio, which is a species with coral pathogens. So when looking at healthy samples, the disease resistant, or the red color morph, and the disease susceptible, the orange color morph, have very different bacterial communities. And the orange morph is dominated by potentially pathogenic Vibrio species. And since bacterial communities play such a large role in health and disease resistance, this difference in bacterial communities may be what's driving some of these differential susceptibility that we see in the field. So that was for just looking at the healthy color morphs. Next, I wanted to see what happens when these color morphs develop disease. So on this plot, we have the healthy red color morph. And I found that the bacterial communities changed significantly with both chronic, the slow-moving form of Montefiore White syndrome, and the acute, fast-moving form of Montefiore White syndrome. And you can see there's no overlap between the healthy and the disease samples. So if we look at what's changing here, there's two major kind of things happening in the bacterial communities with disease. One, we're accumulating lots of Vibrio. So those healthy samples in the red color morph had very little Vibrio, less than 5%. But now with both forms of the disease, we're up to close to 40% Vibrio. And then second, the red color morph is losing bacteria that used to be prevalent. So Listonella, Bacillus, Shimiard examples. So it's losing previously abundant genera and being overwhelmed by Vibrio. So this pattern of changes in your bacterial community with disease is consistent with what's been found in other coral disease systems. So this was kind of an expected result. But for the orange color morph, I found a different pattern. So again, these are the orange samples that were healthy. I found that the bacterial communities didn't shift very much with chronic, the slow-moving form of Montefiore White syndrome. And in fact, they're not statistically significantly different. For the acute, fast-moving Montefiore White syndrome, we did see some changes in between the healthy and the disease samples, but this shift is not as big as what we saw in the red color morph. So if we look at what's changing or not changing in the orange color morph, well, the orange color morph already started out with a lot of Vibrio. So with both chronic and acute Montefiore White syndrome, the abundance of this genus didn't change very much. In acute Montefiore White syndrome affected samples, we are losing some of the previously dominant genera, but we're getting an increase in Hydrobacter, which is interesting. There's not a lot known about this genus. So it could be taking advantage of the dying coral tissue, or it could be a potential pathogen that we might want to investigate in the future. So kind of to sum up the sequencing data, the orange color morph, the disease-susceptible color morph, has bacterial communities that are already in a state of what would be called dysbiosis, meaning that there is an overrepresentation of potentially pathogenic bacteria in the community associated with it. And if this is the case, then you might also suggest that Montefiore White syndrome is acting like an opportunistic infection where there's pathogens already present on the host that are able to take over after some kind of disturbance. So this is similar to Staphylococcus infections in humans. We all carry Staphylococcus on our skin, but none of us get infected, usually unless you're severely immunocompromised or introduce the bacterium via a bad wound or something. So if there are potential pathogens already on these corals, for my second objective, I kind of wanted to confirm that, and then also screen other sources in Kaneohe Bay, so the seawater or other organisms for R3 strains that were established as Montefiore White syndrome pathogens. So to remind you what those were, we have Vibrio corlyticus, strain OCN008, Vibrio owensii, strain OCN002, and a species of Pseudo-Ultramonas, we have the strain OCN003. So in most of my slides, I'm going to be referring to them as their strain name. So I use a combination of culture-based and molecular-based techniques in order to identify these three bacteria in samples I collected. So all three of our pathogens are marine bacteria. They can tolerate salt conditions. So for all my samples, I would first put the sample on a medium that has lots of salts. So in this case, GASW medium. Two of our pathogens are Vibrios and one was not. So to differentiate between those two, I would patch my selected bacterial isolates from here onto TCBS medium, which is selected for Vibrios. If an isolate did not grow on TCBS medium, then it was not a Vibrio. So I would then screen it via PCR with primers specific for pseudo-ultramonas, strain OCN003. So if I got a positive band here, then I would save this isolate to investigate later. Now, okay, so if we go back down this pathway, if an isolate grew yellow as opposed to, say, green on TCBS medium, this is consistent with both Vibrio coriolyticus and Vibrio owensii growth on this media. So isolates that grew yellow, I would screen further. In the case of Vibrio coriolyticus OCN008, it actually produces an antimicrobial compound that it excretes into the surrounding environment, and that actually inhibits the growth of bacteria around it. And if you, on a plate medium, grow OCN008 with other marine bacteria, it can inhibit the growth, resulting in this halo around the dot of Vibrio coriolyticus. This is called a zone of inhibition. So this is a pretty unique property of OCN, or a useful property of OCN008 that is not found in Vibrio owensii OCN002. So Vibrio isolates, I would test to see if they created zones of inhibition against a specific marine bacterium. And if they did produce a zone of inhibition, then I would test them for a gene called ADMP, which is present in the genetic operon that creates the antimicrobial present in Vibrio coriolyticus, OCN008. If a Vibrio did not produce a zone of inhibition, then I would screen it via PCR with primers semi-specific to Vibrio owensii strain OCN002. And I say semi-specific because other Vibrios that I used to test the specificity of these primers did yield PCR products. But in the case of OCN002, I got this double banding pattern that I didn't see in other Vibrios that I tested. So I used this as an identifying marker for OCN002. So this was how I went about looking for these pathogens. And these were the types of samples that I collected to look for these pathogens. I looked at abiotic sources, such as seawater and marine sediment. So every 3 months for a year, I collected seawater from the north, central, and southern regions of Kaneohe Bay. I also screened stream water and stream bed sediments. So once a month for a year, I collected water and sediments from 6 streams that flow into Kaneohe Bay and screened them for pathogens. And then I screened several biotic potential reservoirs. So I looked at the coral itself, Monteverde capitata. I looked at coral-associated organisms such as pinchbeak shrimp, feather duster worms, and blue-spotted goby. And then I also looked at a couple of coral predators. So this butterfly fish will eat polyps, and this is a parasitic flatworm who will also eat that coral tissue. So in the case of the 2 fish, I screened just the surface mucus layers. And for the other organisms, I actually took whole tissue to screen for our Montegrowite syndrome pathogens. So for sources of isolates that resembled Pseudo-Ultramonas, OCN003, I only found this strain in seawater. But I found it in every sampling of seawater they took over the 1-year period. For sources of isolates that resemble Vibrio coriolyticus strain OCN008, I found it in almost everything that I screened. I did not find it in seawater or the parasitic flatworms, but everything else. So one of my samplings of stream water, in this case Kaneohe stream, I found an isolate. I found isolates in both healthy and Montegrowite syndrome affected Monteverde capitata, and all of these other coral reef associated organisms. I did not find a good positive result for Vibrio NCI OCN002. But now that I have all these other isolates, I wanted to investigate them further. So I looked at I did a genetic analysis called multi-locus sequence analysis, and then I also tested their pathogenicity. So first we'll start with isolates that resemble Vibrio corlyticus. So the multi-locus sequence analysis showed that the isolates that I screened from Kaneohe Bay, all of these up here with their own OCN number, clustered most closely and therefore was most similar to the type strain, OCN008, as opposed to other strains of Vibrio corlyticus that have been isolated in other regions of the world. I then took kind of representative isolates, one from each kind of sampling type that I found it in, and exposed these isolates to healthy fragments of Montipora capitata, and I found that they were all inducing acute tissue loss in the laboratory in a time frame and in a manner similar to the type strain, OCN008. For the Pseudo-Ultramonas-like isolates, again, multi-locus sequence analysis showed that they were most similar to the type strain, OCN003. And a representative isolate from one of these I also exposed to healthy fragments of Montipora capitata, and it was also able to induce acute tissue loss in the time frame and in a manner similar to the type strain, OCN003. So I was able to find isolates in the Kaneohe Bay environment that were genetically similar to and genetically similar to the type strain and pathogenic to Montipora capitata, suggesting that these are, I was able to find our Montipora white syndrome pathogens in the environment. I found Vibrio corlyticus in all kinds of things, especially other organisms, but I only found Pseudo-Ultramonas in seawater samples. So because they were only isolated from different samples, this may suggest they have different strategies for infecting Montipora capitata. In the case of Vibrio corlyticus, I found it on the coral host itself. So it could be a pathogen that's already present and able to cause infection when some disturbance happens, like we hypothesized in Objective 1. But Pseudo-Ultramonas may be in the seawater and then can only cause infection after a much larger disturbance, maybe a lesion already present or a wound. So with all of these pathogens and many different sources, it's probably likely that Montipora capitata is constantly exposed to its pathogens. But if it's constantly exposed to its pathogens, why isn't it always getting sick? I mean, they're not all dead yet. So that suggests that there's probably some environmental cofactor that is kind of triggering disease outbreaks, or at least making it easier for these pathogens to get into the coral tissue and infect. So like I said, our outbreaks were associated with heavy rain events, and there's a lot of stressors that can come in with heavy rain events. So a lot of terrestrial sediment just lands on the coral, and the coral has to spend energy to get rid of it. The fresh water lowers the salinity. If the corals are shallow enough, you can get pollutants and chemicals running in from land, and the water itself is quite cold. There's a lot of variables to look into, and if we wanted to do all of them, that would take much more time. I'm just going to present data on one factor that I looked at, and that's lowered salinity, so the freshwater component of the stressor. So in the laboratory, we have to put a lot of bacteria on our coral in order to get tissue loss. In the case of Vibrio corlyticus, OCN008, we have to put 100 million cells per milliliter of seawater in order to get acute tissue loss in the lab. And that would happen within 12 hours to 5 days. In the case of Vibrio owentii, OCN002, you gotta have 10 million cells per milliliter of seawater in order to cause a diffuse tissue loss. And that would happen within average of 28 days post-infection. So I wanted to see if a lowered salinity stress would reduce the amount of bacteria that's required to cause infection, or in the case of Vibrio owensii, if it would lower the amount of days it took in order to cause infection. So the salinity in Kaneohe Bay normally is 35 parts per thousand. And during really heavy rain events, it can dip down to as low as 15 parts per thousand. But that exposure usually will kill a coral fragment very quickly. So I wanted to find a stress that mimicked what would happen during a normal rain event, but would not cause it would non-grossly affect the visual kind of health of the coral. So I chose a 20 parts per thousand stress over the course of 24 hours. So in the lab, I set up these manipulative studies where we had a coral fragment in each one of these aquaria. I would assign coral fragments to a non-stressed group or a stressed group. So the non-stressed group was just kept at ambient salinity, so 35 parts per thousand. But the stressed group got the 20 parts per thousand stress for 24 hours, and then were brought back up to normal salinity. And then after they were brought back up to normal salinity, I would inoculate them with a pathogen. Some of the corals got the normal or high dose of pathogen, so 10 to the 8th cells per mil. Some fragments got an intermediate dose of 10 to the 6th cells per mil. That's a twofold reduction. Some fragments got 10 to the 4th cells per mil, another twofold reduction. And then some fragments were not exposed to bacteria. And then I would wait to see who survived and who didn't. And then this is kind of how that data would look like. So this is % survival over time. So coral fragments that didn't die, you would see a line at the top. Or a group that didn't have any mortality, you'd see a line across the top. So for coral fragments that received no bacteria, whether they were stressed or not, they all survived till the end of the experiment. In the case of Vibrio corlyticus, the high dose, we got mortality in the expected pattern, whether it was stressed or not. So high mortality within 5 days, and this was for both of our groups. For the intermediate dose, none of the non-stressed corals died. But the stressed corals started showing mortality after about 16 days post-infection. We had 50% of them died. For the low dose, none of them died. So in the case of infection with Vibrio corlyticus, the salinity stress did not change the time to infection, at least at the high dose, but it did reduce the amount of bacteria that was needed to cause infection. So I did these experiments again, but with Vibrio owensii, OCN002. Again, the coral fragments were not exposed to any bacteria. They all survived. The non-stressed corals suffered diffuse tissue loss in a manner that was expected. Some mortality after 27 days on average. So this is what was previously reported when we first kind of looked at the strain. But the stressed corals had a higher rate of mortality much quicker, sometimes as soon as 5 days, but on average, 17 days post-infection. For the intermediate dose, all of the non-stressed corals survived, but the stressed corals started showing some mortality after about 16 days post-infection. And then the low dose, they all survived. So in the case of infection with Vibrio owentzii, the salinity distress reduced the amount of time it took to cause infection at the high dose, and it allowed infection at a lower dose. So in this case, the lowered salinity enhances Montebrowite syndrome infection in the laboratory, possibly by reducing the coral defenses, because the pathogens themselves were not ever exposed to the lower salinity, only the coral itself. I would hypothesize that they're disrupting the microbial community, but we'd obviously need to do more experiments to show that. I would love to be able to do that in the future. All right, so in summary for all of my objectives, the disease-susceptible corals, the orange morph, they have bacterial communities that could be considered in a state of dysbiosis, with lots of bacteria that could be pathogens, setting it up for disaster, if you will. So a small disruption makes it, or increases the risk of infection. Montebrowite capitata is probably constantly exposed to bacteria that can infect it. Certainly, there are multiple sources of R3, two of R5 strains of Montebrowite syndrome pathogens. And environmental stress, in this case, lowered salinity, increased the susceptibility to disease for both the pathogens by lowering the amount of bacteria it takes to cause disease in the first place. All right, so with all that, I'd like to thank everybody for being here. I'd like to thank my committee, my two advisors for all of their support and input over the years. I want to thank all my lab mates and all my field buddies and lab support. I especially want to thank Dave Krupp, who is not here, but he let me use lab space out of the community college. It was very helpful. And of course, I want to thank all my friends and family who are here, or those who aren't here, for supporting me and encouraging me through this graduate career. So with that, I would be happy to take any of you guys' questions.
Questions for me? Yeah.
Go ahead.
You mentioned that you did the inoculation after submitting them to this salinity reduction. Then you kind of hypothesized what might be happening to the community on the coral itself, for that susceptibility. But do you have any theories about what's happening to, so you mentioned especially with H008 that it's in the water with them all over, you're shaking your heads.
So Pseudo-Altomonas was the strain that was in the seawater, and that Vibrio coralliticus is potentially some copies of it, I guess, or some cells are already on the coral host.
So for the, I guess for either of them really then, what do you think is happening to those communities of the infectious agent while salinity is changing with whatever is causing the change in salinity in more of a natural environment as opposed to a lab setting?
Right, so the reduced salinity is going to have an effect on the coral tissue itself. This reduced salinity could also have an effect on the bacterial communities in the mucus layers that's going to be exposed to the salinity first. So you could be reducing or inhibiting the growth of some bacteria or bacteria that are able to tolerate lowered salinity. So Vibrios are known to be brackish water bacteria that they can tolerate freshwater fluxes quite well. That helps answer your
Yeah, exactly what I was thinking.
Okay.
So it looks like the red morph of Montebrowite is selective in the bacterial communities it associates with, whereas the orange morph is very promiscuous.
Okay, that would be an interesting way to put that. But certainly, yeah, there's not as much consistency with the orange morph.
Why do you think? Why? Because presumably same species, they're probably, like individuals, are probably reproducing with each other.
Right. So the only other major difference that we know about between these two polymorphs that I didn't mention is that the symbiotic algae that lives inside of the coral and is responsible for producing food for the coral. There are different types of that algae, and these two color morphs actually will harbor different strains of that algae. So we haven't directly tested this, but you know, if you have a change in the food that the algae make for the coral, that may eventually down the line impact the bacterial communities. Some people have started down this route and noticed some differences in bacteria depending on what type of algae is in a coral, but we haven't applied that to this system yet.
Have they ever considered the abundance of carpet pods, given that carpet pods associated with Vibrio?
Right. I did do one screening of copepods. I didn't find anything at first, and I was actually just having trouble growing up bacteria from the plankton samples that I was collecting, so that's kind of why I didn't pursue it further. But yes, Vibrios are known to be attached to.
Because Viviparous can heterotrophically feed on
the
some of the zooplankton. So just kind of made me think of how much we want to look into it.
Did you examine the feeding copepods between the red and the orange?
I did. So that's a whole component of my research that was not presented here. And as far as feeding on brine shrimp, I didn't see a difference between the two color morphs and their rate of heterotrophy in a healthy state. So that may not influence disease susceptibility in this case. Anyone else?
No other questions? Thank me, or thank me. Join me in thanking Amanda, but first I want to mention that we're going to have a reception outside after Amanda's done with the private defense. Where is that, Chris? Right up on the courtyard. In the courtyard on the first floor. Okay, so we'll have some food, we'll have some drinks. Come please help us hopefully celebrate.